My name is Kenneth Witwer, and I'm affiliated with the Johns Hopkins University School of Medicine. I have been studying extracellular RNA and extracellular vesicles for most of my career, and that is because I work on retroviruses like HIV, the human immunodeficiency virus, that is really just an extracellular vesicle that has been hijacked by a foreign genomic program. So when the cell is infected with HIV, HIV integrates into the cell nucleus, and it directs a program that will result in production of progeny. So the HIV virion, that is the virus particle that contains the HIV genome, is an extracellular vesicle, and it contains extracellular RNA. This extracellular RNA includes the HIV RNA, so the genomic RNA. There must be 2 copies of the RNA genome for the virus to be replication competent, but it also contains elements of the host RNA transcriptome, including, quite crucially, a tRNA that is needed to prime the reverse transcription of the virus when it reaches its recipient host cell. My work in ERCC stage 2 has a much broader focus than just the HIV studies, although they will certainly inform what we do. In our HIV work, we have a need to separate virions from other, I would say, host extracellular vesicles. And we do this through a combination of size-based and density-based approaches to separate the particles. In this study, we will be separating other kinds of particles. So different types of extracellular vesicles, but then also lipoprotein particles, which have a very wide range of densities and sizes, and then also ribonucleoprotein particles that carry RNA, the extracellular RNA that we want to learn more about. So we have a bit of a broader and larger goal here with this project than simply the separation of HIV from other extracellular vesicles. The question here is, how does my research look at the ribonucleoprotein particles that in many ways have been understudied in the past compared with extracellular vesicles? So of course we have a lot of interest in extracellular vesicles because there's the chance to trace those extracellular vesicles back to a cell of origin. So the EVs that come from a neuron, from an astrocyte, from an endothelial cell, we have a chance to actually understand their origin by looking at their cell surface markers. But extracellular vesicles actually seem to carry only a minority of the total RNA that we would find in any given biofluid. And we know that the ribonucleoprotein particles really have a wealth of information that is contained in them. And there are likely multiple types of these particles that we can isolate from human biofluids in order to understand the state of health of different tissues and cells in the body. So the ribonucleoprotein particles, one type that has been studied quite a bit, or more than others, is the argonaute microRNA complex. So the argonaute is a protein that contains a microRNA within it. The microRNA is actually loaded as it's loaded into the argonaute during the biogenesis process, during the processing of the microRNA from an immature microRNA to a mature microRNA. And so that complex can be found, for example, within our blood at fairly high concentrations. But this is only one of many complexes. And so I am very excited to have this opportunity to study more of the ribonucleoprotein complexes that are present in biofluids, because I believe that there is a much greater source of information in these biofluids than what we have studied so far. And that would consist of what we could call the degradome. So the degradation products of longer RNAs that have been released, especially from dead or dying cells, that give us a snapshot of what is happening in those cells. And the reason that I say that there's more information there is that the entire transcriptome is constantly being processed, being used, and being degraded. And when this transcriptome, or these degradation products of the transcriptome, end up in our biofluids, we can learn more by examining that information. And so I'm very privileged to be working on my team with one of the first people to recognize the importance of that degradome, and not just of the mature microRNAs, in understanding the health of the individual who was able to contribute a particular biological sample. So we're very much looking forward to looking at what we have previously considered to be junk, but which is actually a treasure trove of information about the health of human beings. Do we plan to identify new RNPs? I think that that's a strong possibility. And with the combined resources of the ERCC Stage 2, and with all the different expertise of our investigators, I believe that we will identify new RNPs, new RNP cargoes that are going to be important in understanding human disease. So at the end of this ERCC Stage 2, I hope that we have accomplished perhaps 3 things, both in my program and then in the program in general. One of those, I think, is to identify ways to better separate these RNA carriers so that we can identify the unique information that is associated with each carrier. A second thing that I would really like to do is understand the identity of the cells of origin of some of these particles. I think that's especially promising with extracellular vesicles, but perhaps it can be done by looking at the cargo alone. And then the third thing I think is that we want to understand and communicate to the clinicians that will eventually be using these assays that we are attempting to develop. What are the conditions that have to be met to make sure that the samples we collect are going to be usable in interrogating the health of our patients? So I think that those 3 things are at least part of what we would like to achieve during this ERCC Stage 2.